Imagine a world where disease is deleted like a typo in a document. A world where suffering is corrected before it begins. Where genetic mistakes are erased with precision. This is no longer imagination. This is happening now. Imagine blindness prevented before birth. Cancer stopped before it forms. Inherited suffering rewritten at the molecular level. The blueprint of life, carefully edited. Humanity standing at the edge of biological power. For decades, genetic diseases were permanent. A single mutation could shape an entire life. Families carried invisible burdens across generations. Medicine could treat symptoms. But it could not rewrite the code. Then scientists discovered something extraordinary. A hidden biological defense system inside bacteria. A molecular tool shaped by evolution itself. It could recognize invading viruses. And cut them apart. That system became known as CRISPR. A simple acronym. But a revolutionary discovery. A tool capable of cutting DNA with precision. A pair of molecular scissors. Think of DNA as a document 3 billion letters long. Inside every cell of your body. Each letter carefully arranged. A single typo can change everything. One error can cause disease. CRISPR works like find and replace. Scientists design a guide molecule. It searches the genome with accuracy. It locates the exact faulty sequence. Like GPS for biology. Then comes Cas9. The cutting enzyme. It moves along the DNA strand. It stops at the marked address. And makes a precise incision. The cell detects the break. It activates emergency repair systems. Molecular machinery rushes to the site. The damaged gene is removed. A corrected sequence can be inserted. For the first time in history, humanity can rewrite its own code. Not over millennia. Not through slow evolution. But in weeks. Evolution is no longer passive. It is programmable. Compared to older genetic engineering methods, CRISPR is shockingly efficient. What once took years can now take weeks. What cost millions can now cost thousands. Laboratories across the world adopted it rapidly. A biological revolution had begun. Clinical trials began targeting blood disorders. Sickle-cell anemia showed remarkable results. Patients once dependent on constant treatment saw improvement. Their own cells were edited and returned to their bodies. For some, symptoms nearly disappeared. Cancer therapy entered a new era. Immune cells were extracted and reprogrammed. CRISPR modified them to better recognize tumors. They were sent back like trained soldiers. Targeted. Precise. Deadly to cancer. Agriculture transformed as well. Crops were edited to resist drought. Plants became stronger against pests. Food production became more resilient. Genetic precision replaced random mutation breeding. Scientists even explored eliminating inherited blindness. And preventing certain heart conditions before birth. The potential seemed limitless. Every disease looked like a puzzle to solve. The genome became a map of possibilities. But with power came unease. Editing adult cells was one thing. Editing embryos was another. Because embryo edits are permanent. They pass to every future generation. The stakes suddenly became generational. In 2018, the world was stunned. A scientist announced the birth of gene-edited twins. Their DNA had been altered as embryos. The goal was resistance to HIV. The announcement triggered global shock. The scientific community reacted immediately. Many called it reckless. Ethical boundaries had been crossed. Regulations had been bypassed. Humanity had stepped into uncertain territory. The issue was not whether CRISPR worked. It clearly did. The issue was control. Who decides which traits are desirable? And who prevents misuse? Because once a gene is changed in an embryo, it echoes forward. Through children. And grandchildren. Evolution is no longer blind. It becomes intentional. And intention can divide humanity. Even with precision, CRISPR is not perfect. Sometimes it cuts in the wrong place. A tiny mistake in billions of letters. An unintended mutation. Invisible at first, but potentially dangerous. These are called off-target effects. Small changes that ripple through the genome. Most are harmless. Some are not. Biology is complex beyond prediction. Researchers are improving accuracy every year. New variants of Cas enzymes are more precise. AI now helps design safer guide molecules. Technology is evolving alongside biology. But the ethical debate grows louder. Should we only treat disease? Or enhance intelligence? Strength? Appearance? The line between therapy and enhancement begins to blur. Imagine a world of designer genetics. Parents selecting traits like options on a screen. Height, eye color, cognitive ability. Evolution guided by preference. The risk is not just biological. It is social. Access may favor the wealthy. Genetic inequality could deepen. Humanity could divide in new ways. Yet the benefits are undeniable. Children free from inherited suffering. Families spared generations of pain. Diseases erased from the human story. Hope written directly into DNA. For billions of years, evolution was shaped by chance. Random mutations. Natural selection. Slow adaptation. Now, intention enters the process. CRISPR is not destiny. It is a tool. Tools reflect the hands that use them. The future of our species is not written in genes alone. It is written in choices. If you could erase one genetic disease forever, would you? Where should the boundary be drawn? At healing? Or at enhancement? The next chapter of evolution may be decided by us.